Good evening, everyone. Thank you for your. Can you hear me?
Yeah.
Okay. Thank you for your kind introduction. Today I will talk about SCERPA, which is a tool basically that works on the field of molecular field-coupled nanocomputing. I will start with a brief presentation of what molecular field-coupled nanocomputing is. I think you already know about the Moore's law and the problem of CMOS scaling. Molecular field-coupled nanocomputing is, let me say, a new technology proposed in the field of beyond-CMOS, intended to overcome many of the problems of CMOS scaling. Basically, it's a nanometric technology based on molecules. In this slide, you can see the so-called dispersing molecule, which is a molecule ad hoc synthesized for this technology. If we consider two molecules encased in the so-called molecular SCN cell, and we oxidize these two molecules, what happens is that we have basically an electrostatic repulsion between the two molecules. That basically forces the two charges, because we have oxidized molecules. That means we have removed one electron, we have positive molecules. What happens is that we basically position the two charges on the antipodal sides of this molecular cell. The interesting point is that basically we have two possible configurations that are, from an energy perspective, stable, and we can associate an information to the two states, so logic 1 and logic 0. So we have a way to encode the information that basically does not exploit any voltage or current. Additionally, generally we have also a third state, because if we juxtapose the two molecules, and we may suppose that we have a certain charge distribution encoding a certain logic, what we can do is to apply an electric field that basically pushes the two charges to go in the bottom part of the two molecules. That is kind of a reset state, and then we talk about reset field or clock field. This is needed to erase the information, or in some cases if we want to, for instance, avoid the propagation or the elaboration information in some of the molecules. And then, in this example, we have the propagation of the information in the so-called molecular wire, where basically we have a series of molecular cells, and we divide this simple circuit in clock regions. And we have the possibility, by exploiting what I just mentioned, the clock field, to activate each region in an independent way. If we activate the first region due to electrostatic repulsion, so Coulomb repulsion, we have the propagation in the first clock region. Then we activate the second and the third. And what happens is that basically we have a way to propagate the first logic encoded by the first cell, that we generally name driver, to the output of our molecular wire. This is a way to do the propagation of the information, but by using specific patterns, we also have the possibility to do some information elaboration. For instance, on the left of this slide, we have the inverter, that basically, using the electrostatic repulsion between molecules, allows for to invert the logic in the device. So if we have a 0 in the input, we have a 1 in the output, and vice versa. So we have the inverter. On the right, we have the majority voter that we will see in the results of this presentation, but basically encodes at the output of the device the majority of the inputs. For instance, in this case, we have two red inputs and we have one blue inputs. That means at the output we will have a red output. These two logic devices, together with the wire that basically propagates the information, allows for a complete set that basically permit to create any digital device by using only the inverter and the majority vote. So what are the advantages and why molecular SCN is promising? So basically, first, we can see that there is no electrical transport. We have no movement of charges between one molecule and the other. That means very low power, because we have no current. You know that the current basically determines the power dissipation in CMOS circuits. Here we have no current, so the power dissipation is very low, and we are beyond the Landauer limits in many cases. We have the possibility to work at ambient temperature. Many of the beyond-CMOS technology, especially when we talk about nanotechnology, require cryogenic temperatures. here we have a technology that has been supposed to work at ambient temperature, so room temperature. High speed, so this technology is supposed to work at the terahertz frequency, so very high speed, and self-assembly. So basically, it's very simple to fabricate uniform layers of molecules which are uniformly sized. That does not mean it's easy to create a device, but it's easy to at least create a uniform layers of molecules. So it's very simple to control the specific dimension of each device. But what is the problem? And especially, what is the problem addressed by this research? The problem is the simulation. You know, when you do design of digital circuits, the simulation of devices is fundamental, basically, for the verification, for the simulation, and for the design of digital circuits. The problem with molecules is that the equation that you need to solve to evaluate any property in the molecule is the Schrödinger equation, which is very difficult to deal with. On the left of this slide, we have some possible analysis that we have performed using ORCA, that is basically a solver of the Schrödinger equation. And if you see the last line of the log file, what you read is 2 days, in the second case 16 hours, or even almost 50 days. And this is the analysis of a single molecule. Of course, if you want to simulate an entire device, you may understand that these times are not feasible, because if you just do 50 days times 1,000, you may understand that it's very difficult to do a simulation. And this is why we have here at Politecnico di Torino discussed and introduced a new methodology, that is the mosquito methodology, that allows you to simulate and to consider molecules as electronic devices. It consists of basically 3 steps. The initio characterization, that is basically we solve the Schrödinger equation only to evaluate the property of a single molecule, no more than one molecule, only one molecule. We have the extraction of figure of metrics that basically allows us to describe what is the distribution of the charge on the molecule according to the electric field on the molecule that may be generated by external electric fields or, external electrodes or other molecules. And then we have a tool that is what we are going to describe deeply in this presentation, that is SCERPA, and allows you to simulate the entire circuit by using the mosquito methodology. So here I'm going to explain what I need with a molecule as an electronic device. If we have a very small wire composed by 3 molecules, and we consider, for instance, the molecule in the center of the wire, this molecule will be subjected to the field generated by the other molecules. That means we may evaluate an input voltage by integrating the field, for instance, on the ferrocene of these specific molecules. Then, if we have somehow characterized with ab initio technology, with ab initio techniques, the charge distribution of the molecule, we can associate a certain charge to the molecule by looking at the voltage. And then, if we have the charge distribution on this molecule, we can also evaluate the field generated by this molecule on the other molecules, and then associate a new charge distribution according to the field using, again, the transcharacteristics. So, basically, the working principle of the SCERPA algorithm is to create a self-consistent procedure on this concept. So we analyze all the molecules of the device, we evaluate the field generated by all the molecules on a certain molecules, and we associate to this voltage a certain charge distribution according to the transcharacteristics obtained with ab initio techniques according to a certain clock field. Currently, the algorithm, the SCERPA tool, is subdivided into 3 parts: the layout, the algorithm, and the viewer. And we have basically 2 parallel paths for this tool because it is available in 2 different versions. A first version that is basically for development and research applications, it's based completely on MATLAB. Whereas the second version that is still under working, it's based on MATLAB for the layout and on the Topolino suite for the simulation and for the viewer. These versions tend to be compatible one to the other. So for instance, we can start, and today we will see some layouts generated by MATLAB, but then we can perform the simulation and the viewer analysis using the MATLAB scripts. So the first part of the SCERPA tool is the layout. Basically, as I said, we can have two possible layout editors. The first one is MATLAB, that basically we just write the structure of the device using matrices and some values. Or we can just design our device on the market tool, which is a graphical tool to create device. It allows us to perform a lot of configurations. For instance, we can determine the layout, we can define what are the clock regions, we can determine what are the lattice parameters, so we can, for instance, decide what is the intermolecular distance, or we can add some process variation, we can add shifts, rotations, we can add a lot of other process variation like the substrate roughness. We can also employ some finite element methods. And basically, we can also define what is the test bench of the circuit. So we can determine the input, the output, the clocking signals, and eventually also the external voltages. One interesting point of our layout editor is the fact that we can implement devices with more than one molecular type. So basically, we can decide to put on the same circuit different molecules, because molecular SCM can be performed with different molecules. We saw the dispersing, but we have other molecules. In this case, for instance, we tried to, and we succeeded in implementing, for instance, a neuron. So where, in this case, we basically created a majority voter where the inputs are somehow weighted in the transcharacteristics of specific molecules. So each of the input of the majority voter here is functionalized with specific transcharacteristics, and we are able to create a neuron based on molecular SCM technology. So we have the algorithm section that basically performs all the calculations related to the charge distribution and to the field generated by the layout. And this is, of course, the logo. It's basically a self-consistent procedure. We are able to tune the simulation by varying, for instance, the convergence thresholds. And we can also export some data in order to be available on the viewer. Or we can vary basically the interaction radius and active region thresholds. That basically allows you to, let me say, optimize the calculation, because the idea is that when you evaluate a certain part of the circuit, it's not necessary to consider all the intermolecular interaction independently on the distance. So very far molecule will not interact, and if you have the interaction only on a certain part of the device, it's completely useless to evaluate the interaction also in other parts of the circuit. So we have some optimization techniques that basically allows you to optimize the calculation. And finally, we have the viewer. Here we have, for instance, the waveform viewer associated with the wire, and we can see, for instance, the first three lines represents the clock fields associated to the three clock regions, so yellow, green, and blue on the layout of the circuit. And we can also see in red the input of the device. Basically, here we just have a very simple transition from 1 to 0, and we see in blue 1 to 0. That means we have propagation of the information from the input to the output. What is interesting in the SCERPA viewer is that basically you can extract the so-called QSS file. That is the one you see on the right, and you can analyze a single step by using other types of plots. And basically we have four possibilities to view the single step. The first one is the 1D charge viewer, that basically allows you to measure the charge on a single molecule. So you just see each molecule with the associated charge encoding the logic. You see an alternation basically because there is an alternation of the wire that is typical of molecular SCM propagation. We have the 3D viewer that is not very easy to see from here, but basically shows you all the molecules. You can see a geometrical representation of the molecule with some spots that basically represent the charge on the molecule. We have the potential viewer that basically represents kind of a measurement of the potential of the device. You may imagine to have a tip that basically moves on the device and measure the voltage on each point of the space. And You see the white spots that are basically a representation of the alternation, the charge alternation, typical again of the molecular propagation. And finally, we have the logic viewer. That is not very relevant for a viewer, but we will see in a few slides it will be more useful for complex devices. That basically allows you to associate a certain color to the potential viewer or the charge viewer. So if you see red, for instance, it means that you are propagating zero, a logical zero. So I will show you some possible results. In this slide, we have again the majority voter. We see that we have two types of majority voters. The first one is the classical majority voter that you may find in the literature. In the second one, in the second part of the slide, we see an enlarged version of the majority voter that is basically more robust. So we designed this majority voter in order to be less sensible, for instance, to the substrate roughness. If we analyze this device using SCERPA, here we can see the logic viewer that basically shows you how the majority voter was. And if you see all the steps, you will see that the output, that is the, let me say, the branch on the right on each of the 8 figures, is of the same colors of the majority of the input. So if we have more zeros, we have 0 at the output. If we have more ones, we have 1 at the output. And it was in both the circuit layout. So it is possible to create a majority voter using a single line device or a multi-line device, which is supposed to be more robust to substrate roughness. This is the inverter. Again, here you see the logic viewer, and you see that it's red in the input, at the input, and blue at the output. That means we are inverting the propagation of the information. We can see that the same plot can be seen both logically or physically. So here we can see the white spots representing the voltage on each of the molecules composing the circuits. And I'm not sure you can see from far, but what you see is that the zigzag that you see on the potential viewer is basically inverted in the input and in the output, so associated to the red and to the blue colors. One thing that we always say about the SCERPA tool is that it allows you to link the physical behavior of the molecule to the circuit behavior. Here we have, for instance, an inverter that may be found in the literature that has been proposed for general QCA computation, quantum cellular automata. It is basically based on a single branch. So we have no double branch as we saw in the previous slide, but we have only a single branch. What is interesting to see is that according to the type of molecule, this device may work or may not. And this is very important because in many cases in the literature it's just taken for granted that the device works. But if you analyze with SCERPA, you will understand that this may work only in the case that the molecule is neutral. So we do not have like a single electron encoding information, but we have a positive charge and a negative charge on the same molecule. Or if it's the so-called Zwitter ionic molecule, that means it's oxidized but still neutral because somehow in the molecule we have also a negative charge. So this is very important for us because it's the first demonstration that SCERPA basically allows you to link a physical behavior to the system level behavior of the circuit. So the last result I would like to show you is the XOR device. Here it's not possible for me to show the complete simulation physically, but I just will show the logic waveform. Again, the device is completely working, so we have the behavior of the XOR basically. And again, we can also see the propagation of the information by analyzing all the steps. And this is a GIF, for instance, obtained with the SCERPA algorithm, where you can see the propagation of the information in the device. And then, future perspective, we have a tool that is continuously under development, and we are going to integrate a lot of features in the next months, if not years, which is, for instance, the energy evaluation, both intended like power dissipation and signal energy. We are going to improve the capability of the tool to be in the design framework of Topolino. We are also going to introduce dynamic simulation, so to allow the designer to perform transient analysis. And then we have other features that will be added very soon. So, before concluding my presentation, I would like also to thank all the people that basically works behind the scenes of the SCERPA development, which are basically the people working in the VLSI group, some professor, postdoc, and PhD students, and a lot of master thesis students that worked on the algorithm in the previous years. And of course, I would like to thank you for the attention, and especially to the IEEE Student Branch for this incredible work that I know it's very difficult sometimes. So thank you very much, and thank you very much for the attention.